Hello everyone, I'm Riley and welcome to Fukusaki Laboratory. Now, I would like to explain one of the research topics in our laboratory, food metabolomics. From raw materials to becoming food that we eat, food goes through many processing steps. These steps, such as storage and cooking process, can affect food components that ultimately reflect on differences in sensory attributes and perceived food quality for consumers. Therefore, food metabolomics in Fukusaki Laboratory focuses on analyzing small molecules such as amino acids, sugars, and organic acids to understand more about the food control and production, and therefore can give discriminative, predictive, and informative data. First, let's talk about predictive analysis. Metabolomics has enabled researchers to predict various food phenotypes through statistical models by using metabolome data as an explanatory variable and sensory analysis data from trained panelists as a response variable. Next, discriminative analysis metabolomics involves investigating the differences between sample population without necessarily obtaining detailed information about the sample. On the other hand, for informative analysis, metabolomics can characterize and identify target compounds to search for novel bioactive compounds, biomarkers, and create metabolite databases of specific food samples. This metabolomics approach can be applied to various food products. This ranges from natural food products from agriculture and aquaculture to processed food products such as fermented food. Now, let's talk about studies that have already been conducted in Fukusaki Laboratory. First is metabolomics for fruit ripening studies. Bananas and mangosteens are fruits that can be ripened even after harvest, and color changes are commonly used as ripening parameters. To study about it, we used metabolomics analysis and color measurement as a response variate to correlate the ripening process and metabolite changes. These studies could provide important clues for understanding ripening mechanisms and identify biomarkers that might be useful for post-harvest technology. Not only fruit ripening, but studies of different cultivars or varieties of fruits can also be performed through metabolomics. For example, in this laboratory, we also conducted component profiling of mangoes from different cultivars. We conducted the metabolomics approach and sensory evaluation through the collaboration of an Indonesian research agency to obtain general characteristics of these mangoes. The data can be used to perform discriminative analysis to look for unique attributes of these varieties. The next study is metabolomics for tempeh. Tempeh is an Indonesian fermented food that is made by inoculating fungi with cooked soybeans. So, it is similar to Japanese natto, but tempeh has a solid texture. Throughout the world, tempeh is known as a meat alternative and is a very important source of protein in Indonesia. In Fukusaki Laboratory, we studied the differences between the tempeh production method and analyzed the difference in the metabolome profiles of the cooked tempeh. Using the metabolomics approach, we can discriminate tempeh based on their production procedure by using color data as the response variable due to the darker color in traditional production compared to modern production. In our laboratory, we have also conducted studies related to aquaculture products, and in particular, one study involved investigating the factors affecting texture in shrimp. Now, cooked shrimp itself is a very important commercial product, but what factors affect the final sensory characteristics of this cooked shrimp have not yet been elucidated thus far. By examining the metabolome of cooked shrimp, along with other physicochemical profiles of shrimp, such as performing texture analysis, we can correlate this metabolite profile with texture profile and see which components result in differences in cooked shrimp. One of the more exciting studies that come from our laboratory involve coffee because of the worldwide love for coffee. Now, to determine coffee quality, there is a standard procedure called the cupping test that is conducted by trained and certified panelists. If the coffee is scored more than 80 points out of 100, these coffee beans are categorized as specialty coffee. And to know what the key compounds are that correlate with coffee quality, metabolomics is a powerful tool to answer this question. So, the cupping score and metabolome data can be combined and correlated using multivariate data analysis, and the result from this data analysis could suggest compounds that have important correlations with coffee quality. To confirm the result, validation by additional tests is necessarily conducted. Lastly, I would like to introduce the more recent metabolomic studies that have come from our laboratory, studies that involved the coconut. The coconut itself is a staple tropical fruit used widely across the world and is known as the tree of life for all the various uses of each part of the coconut. And these three studies here looked at the coconut from different angles using metabolomics as the common strategy. First, quality preservation of coconut water during storage. Coconut water itself is a high-value commodity that is usually treated thermally in order to preserve its quality for a long time. However, the flavor of this coconut water, because of the thermal treatment, can be changed. Thus, this study tries to look for methods to preserve the coconut water quality without affecting the flavor through non-thermal treatment. And this kind of treatment can be examined through metabolomics and sensory evaluation to confirm the findings. Next, another study is trying to characterize copior. Copior is a unique coconut phenotype with abundant and soft flesh. And up to now, no study has yet looked at what differentiates copior between other coconuts. Lastly, another study about coconuts in our laboratory observes the influence of origin and variety on the coconut metabolome profile. By looking at different coconuts from different countries and from different varieties, patterns in metabolite profiles between these samples might be observed, and therefore, ideal uses for each type of coconut from each country and variety might be suggested. And so overall, for these three studies, we can see again the correlation of metabolite profiles with both physicochemical and sensory profiles to draw some major conclusions. In our laboratory as well, we have a very long list of publications related with food. And I highly suggest that if you are interested, please go ahead and read some of the publications that we have produced from our laboratory. That is it for my presentation. If you are interested or you might have some questions, you could come to Fukusaki Laboratory directly or contact us on our homepage. Thank you very much for your kind attention.